All right, getting ready for your GCSE biology exams? You've come to the exact right place. Today, we're going to crack the code on one of those key required practicals, the osmosis investigation. This is a big one from section 8.2.3 of the spec, so let's dive right in.
So let's start with the classic, if slightly grim question. Why does putting salt on a slug make it shrivel up? Yeah, I know it's not pretty, but it's actually the perfect real-world example of the powerful process we're looking at today. And the one-word answer? Osmosis.
Okay, so to really get what's happening with that slug, and more importantly with our potato practical, we first need to nail the core science of osmosis. Trust me, getting this part right makes everything that follows an absolute breeze.
And here it is, the official AQA definition you've got to know. Let's break this down, because it's not as complicated as it sounds. Osmosis is really just a special type of diffusion. But here's the key. It's not about just any substance moving around. It's specifically about the movement of water molecules. And they move across a very special kind of barrier. This slide really helps clear things up. When we say a dilute solution, all that really means is there's a high concentration of water molecules. On the flip side, a concentrated solution has a low concentration of water molecules, because other stuff like sugar is taking up all the space. So, at its heart, osmosis is just water moving from where it's crowded to where it's less crowded. Simple as that. And here's the process in action. The water molecules are all buzzing around randomly. But because there are way more of them on that dilute side, it's just more likely that they'll pass through the membrane over to the concentrated side. This creates what we call a net movement down a water concentration gradient. It's all about evening things out.
Okay, theory's done. Now for the fun part. Let's apply all this to the practical itself. Required Practical Activity 3. This is where you get to see osmosis happen right before your eyes and collect data like a real scientist. This is where it all comes to life.
So every great experiment needs a clear aim. And for this one, our mission is to figure out how changing the concentration of a solution affects the mass of some plant tissue. In our case, a good old potato. Is it going to soak up water and get heavier? Or will it lose water and get lighter? Let's find out. Here's the whole method in a nutshell, and you'll notice consistency is everything here. The potato cylinders need to be the same size, you leave them for a set amount of time, you get the idea. You measure the mass before, pop them into the different solutions, wait a bit, then measure the mass again. Now that final step is crucial, gently blotting them dry. You want to make sure you're only weighing the potato, not a bunch of extra water clinging to the outside. And of course, you can't talk about an experiment without talking about variables. First up, the independent variable. That's the one thing you deliberately change, which is the concentration of our sugar or salt solution. Then we have the dependent variable. That's what you measure to see the effect. In this case, the percentage change in the potato's mass. And finally, you have your control variables. All the things you have to keep exactly the same for it to be a fair test. We're talking time, temperature, and the volume of the liquid. So you've done the experiment, you've got a page full of numbers. Now what? Well, this next bit is all about transforming that raw data into actual, meaningful knowledge. This is how we find the story hidden in the numbers. And here's a really important step. We don't just look at the raw change in grams. No, we calculate the percentage change. Now, why does that matter? Well, it's because your potato cylinders probably didn't all start at the exact same mass. Calculating a percentage levels the playing field and gives us a fair, standard way to compare all our samples. This is a massive math skill for your exam. So, a finished results table might look something like this. Check out that top row, the one in distilled water. The final mass is higher than the start mass, so the potato gained water through osmosis. That gives it a positive percentage change. But then look what happens. As the solution gets more and more concentrated, the percentage change turns negative. That means the potato is now losing water. Drawing a graph just makes the pattern jump out at you. This visual shows the trend perfectly. In the dilute solutions, the potato gains mass, and in the really concentrated solutions, it loses mass. It's such a powerful way to show your findings, and it makes the relationship between concentration and mass change totally clear.
Okay, this right here is the big takeaway from your graph. And honestly, this is where it gets really cool. See that point where your line of best fit crosses the x-axis? That spot where there's 0% change in mass? We call that the isotonic point. What it means is that at that exact concentration, the water inside the potato cells is perfectly balanced with the solution outside. So no net water movement, no change in mass. You've basically found the potato's internal concentration.
So, you've done the practical. You get the results. What now? Well, now we focus on turning all this knowledge into exam success. It's time to think like an examiner and get ready for the kinds of questions they might throw at you.
In an exam, they love asking you to evaluate the method, and these points are your secret weapon. For instance, was each potato cylinder blotted dry in the exact same way? What if a little bit of water evaporated from the beakers, making the solutions more concentrated? Being able to spot these potential errors, and even better, suggest ways to improve the experiment, shows a really deep level of understanding.
And to wrap it all up, here's exactly how this practical ticks all the boxes for the AQA specification. Look at that! You've practiced using equipment, making accurate measurements, using a balance with precision, plus you've nailed key math skills like calculating percentages and drawing graphs. So by mastering this one practical, you're actually getting revision done for a whole bunch of skills you'll need in that final exam. So, we started with a slug, we investigated a potato, and we've absolutely nailed the science. My final question for you is this. Now that you've mastered osmosis in plants, where else is this process absolutely essential for life to even exist? Think about your own body, your kidneys, every single one of your cells. The principles are exactly the same. Keep that curiosity going.